Now that we've spent some time learning about bacterial growth, learning about bacterial nutrition, and the kinds of heterotrophs and autotrophs, etc., we've learned a lot about the conditions under which bacteria can survive and conditions they find unfavorable for growth. We want to focus now on what's happening inside these cells. And we're going to be learning about the major metabolic pathways in cells, including glycolysis and the Krebs cycle, the electron transport chain, and fermentation. But before we do that, we want to understand some of the basic principles around metabolism with a special focus on enzymes and how enzymes work. So metabolism is actually a word that encompasses all the chemical reactions that occur in cells. And we can think of two major categories that both fall under the umbrella of metabolism. So the first is catabolism, and this is where usually bigger molecules are broken down into smaller molecules and they release energy. Usually that energy is in the form of ATP. So we have complex molecules being broken down. It releases heat and chemical energy in the form of ATP that cells can use to do work. Cells also undergo anabolic reactions. To undergo an anabolic reaction, energy is required, at least it is in cells. And what you see is small molecules here with the help of ATP are put together in an anabolic reaction to build larger and more complex molecules. And if you think about it, a cell, most cells are going to take up glucose and they are at some point going to divide to produce other cells. So that glucose needs to be not only a source of energy, but also raw materials for proteins and lipids and carbohydrates and all of the complex molecules that cells require in order to make a copy of themselves. And I like this picture for that reason. It shows a glucose molecule here. This is a nutrient that the cell gets from outside, and it can certainly undergo catabolic reactions inside cells. And again, there are major pathways associated with catabolism in cells: glycolysis, Krebs, the electron transport chain, they're calling it respiratory chain here, that's the same thing, and then fermentation. So this yields energy for sure. But another thing that these chemical pathways yield are intermediate molecules that can then be used to build other molecules the cell needs. So pyruvate is the end product of glycolysis. Acetyl-CoA is the starting material for the Krebs cycle. Glyceraldehyde 3-phosphate is an intermediate in glycolysis. And these molecules are all really important in building fatty acids, in building nucleotides to make nucleic acids and DNA and RNA, to make other sugars that the cell may need, just to store glycogen in liver and muscles if you're an animal, and to make amino acids to build proteins. So these precursor molecules that are obtained from breaking down glucose are then what the cell uses to build things like proteins. If you're a bacteria, you'll need peptidoglycan for your cell wall. Organisms need RNA and DNA. They need the complex lipids that are part of their cell membranes. And it's really building all of these macromolecules together are then assembled into a cell. In this case, it's showing a bacterial cell. And all of these are anabolic processes as we take that simple glucose molecule and produces a very complex bacterial cell. And of course, we learned last time that this whole process can take place in about 20 minutes for some bacteria when conditions are good. So it's a phenomenally complex process that happens incredibly rapidly. Now, it wouldn't happen at all without enzymes. And so that's what we're going to focus on next. Enzymes are molecules that catalyze biological reactions. So how do enzymes work? Well, this purple blob here is trying to illustrate an enzyme, and it binds to something called a substrate. So this is the molecule that's going to change, and by binding to the enzyme, the substrate then reacts with the enzyme or interacts with the enzyme in such a way that the substrate is now, in this case, broken down from a larger molecule into two smaller molecules. One key feature is that the enzyme itself is unchanged. So once this reaction occurs, you have the enzyme is regenerated, and it can then participate. It can take many of these red substrates and turn them into products. So this is the fundamental idea behind an enzyme. And cells need enzymes because many chemical reactions that occur in cells would not occur at temperatures and pressures and pHs that are sustainable for life. And yet the cell needs these chemical reactions to happen. So without enzymes, the cell can't survive. And in fact, when we go to try to kill bacteria by sterilizing them with heat or by sterilizing them or sanitizing with chemicals, we often target proteins and it's specifically enzymes, because once you destroy the cell's enzymes, it no longer can survive. We refer to enzymes when we describe them, we always give them the ending ase. So for example, an oxoreductase is an enzyme that participates in redox reactions. Transferases transfer functional groups. Hydrolases add water to break down larger molecules. The enzymes in your gut that help break down lipids and proteins, this is sucrase that breaks down sugar in the form of sucrose. These enzymes all are hydrolases, so they add water and they break chemical bonds by adding a water across the bond. So lipids, proteins, and sugars, these are all important proteins in the human gut. Lyases remove atoms or groups of atoms without water. Isomerases rearrange atoms. Actually, one of the first enzymes in glycolysis is an isomerase. That's a key part of that pathway. And then we have ligases. Ligases join molecules together. Oftentimes when we refer to ligase, we're thinking of DNA ligase, which actually hooks the backbone of DNA together. But there are other examples of ligases. Acetyl-CoA synthetase is a ligase, and it's critical in forming acetyl-CoA for the beginning of the Krebs cycle. So if a protein has the name with an ase on the end, it's probably an enzyme. And you can often tell by the first part of the name, transfer, ase, hydrolase. You can almost always tell what the job of that enzyme is by reading what type of or what class of enzyme it is. So I want to explore enzymes in more detail. And in order to do that, I found two videos which are both excellent. The first one I'm going to post is an Amoeba Sisters video. It's very basic, but it covers a lot of the things that I'm going to be talking about. And so hopefully it will really help you get a sense of the big picture of the topics that we're going to focus on. The second video is more complicated. It's the same information. Actually, it's got a little bit less information, but it really shows what happens in the active site of an enzyme. Don't panic. You don't need to memorize the exact chemical reactions that are occurring, but I do want you to get a sense of how the amino acids that are part of that enzymatic structure, how they interact with the substrates to catalyze these chemical reactions. And the graphics are beautiful. So it's worthwhile watching both videos. And then when you come back to me, we'll go into more detail and explain the key points that are covered in those videos.